Hello, my name is William Antcliffe and I'm doing this presentation on behalf of the Royal Veterinary College as a first year PhD student. Today I'm just going to be discussing a brief snapshot of the work that I've been doing, which is all about the exposure to early life stressors during zebrafish neurodevelopment and the impacts that this can have. It is worth noting that this video presentation is designed to be watched in conjunction with an academic poster that I'll be presenting at the Anatomical Society Summer Conference 2024, which is hosted in Edinburgh. I've put a copy of the full poster on the screen now, but if you're coming here from elsewhere or you've just stumbled upon this video because you're interested in the science that we're doing at the RVC, some of the aspects within this presentation might feel a little bit rushed or it might not make as much sense. And if that's the case, then I've linked the poster down in the description below because this image is a little bit tiny and we're also going to be going through it step by step in the presentation. In the title, we said early life stressors as this general term, but really we're talking about two in particular. And the first one is pollution. Why pollution? Well, because it's literally everywhere. It doesn't matter how you interact with the world, no matter how hard you or I try, we're exposed to it all of the time. And in this study, we're specifically using urban particulate matter. However, the reality is that pollution on its own is likely not enough to lead to more severe changes. Otherwise, we'd be seeing a lot more problems within the existing population out in the real world right now as a result. And so for our second stressor, we're using cortisol, given its ability to mimic elevated maternal hormonal stress levels. Now this is actually quite an important and deliberate combination because there is an inherent link between maternal stress due to poor socioeconomic conditions and poor air quality. With some background on what these stressors are and why they're important, we can start to think about what kind of effects they might have on neurodevelopment. We hypothesised that prolonged exposure to urban particulate matter, or UPM, during early life development, both in isolation and in combination with other stressors, affects zebrafish neurodevelopment. From this hypothesis comes some natural research questions, which you can see on the screen here. Now this section of the poster nicely summarises most of the technical work that I've done so far. The key things to note are that we're using zebrafish as the model organism. Now you'll notice that they've popped up a few times already in the presentation, but I haven't really gone into much detail about why, and that's because I don't have the time to go into the biological background now. All you need to know is that they're a good model for vertebrate development, in particular neurodevelopment, and they're really useful for experimental design too. But to assess neurodevelopment overall, we looked at both structure and function of the nervous system. And what that means is we've looked at detailed images of the brain and recorded lots of behavioural measurements as well. And then we've also dabbled in a bit of new computational analysis in order to better understand the data that we've been getting. I'm going to jump right into the morphometrics now, and this involves taking measurements of the zebrafish that have been exposed to our different stressors. In this case, we're taking all measurements at 5 days post-fertilization, and on the screen now I've just put up a couple of example measurements that we might be able to expect to take at this time point. What did we find when we took these measurements? Well, zebrafish exposed to UPM have reduced head width. The same thing happens to those exposed to cortisol, and then interestingly, those exposed to both UPM and cortisol together showed the biggest reduction in head width. When we look at the head length, however, interestingly, we don't see this same pattern. In fact, it's almost the opposite. However, we didn't get any significant differences when we did this. And then another interesting metric is the distance between the eyes, otherwise known as the interocular distance. We saw no significant changes when exposed to UPM, no significant change when exposed to cortisol, but we did see a significant decrease again when exposed to both UPM and cortisol. And to show you that this isn't affecting all aspects of their morphology, here I've just presented some control factors to go alongside the data I just showed you. The overall length of the zebrafish was conserved, as was the heart and yolk sac areas, all of which are fundamental to general zebrafish development. So just to bring these two avenues together, what this means is that we're seeing changes in the head that are specific to that region. So to put it simply, we saw changes in some regions, but not others. And this suggests that the UPM and cortisol are having a specific effect on development. To track movement, we use a machine called DanioVision, which is an overhead camera that records zebrafish as they move within well plates for a set period of time. For all of our experiments, that time is set to 20 minutes. And again, here's an example of what this looks like at 5 days post-fertilization. We can see the zebrafish in each of the well plates and their path is being tracked in red. And then just on the left over there, I've put a picture of the DanioVision machine, which is the overall machine that actually records this data. So now if we take a deep dive into their overall movement across this 20-minute period, the nature of this being an experiment that involves two stressors being used simultaneously means that we have lots of different groups going up against each other. And so I just want to spend a bit more time walking you through this step by step. We first focus on these three groups. We have our control, which is our system water, with two increasing concentrations of UPM, 30 and 100 micrograms per mil. We can see that there's reduced movement when larvae are exposed to these increasing concentrations of UPM. And in fact, these are significant when analysed using a mixed effect model, which is what accounts for batch variation between biological replicates. But we've got two stressors, remember, not just one. We also want to look at the cortisol and see what that's doing. So just comparing the cortisol group to our control group, our system water, we can see that there's a reduction, but this is not significant. And now if we shift our focus to the right side of the graph, we can compare our multiple stressor groups, which is the UPM and the cortisol. We're first going to compare them to our cortisol only group. We can see that the total distance was reduced. And we get significant differences here. So the addition of UPM with cortisol has a clear additive effect. We also want to compare these same two hit groups with their UPM only counterpart. In both cases, we see that there is a reduction in movement, but interestingly, neither case is significant. And then finally, we can compare these multiple stressor groups with our original control, the system water. And we can see again that there are significant differences between both these groups in comparison to zebrafish raised in our lovely clean system water. But we don't just want to look at distance on its own, we also want to consider other factors as well, such as the maximum velocity. Now, if we apply the same comparative thinking as we just did for the dataset on the far left to the one on the far right, we can see that there isn't quite as much of an additive response as to what we saw with the total distance moved, but we do see that the maximum velocities are reduced across all treatment groups, with those that are exposed to both stressors simultaneously, UPM and cortisol, showing the biggest reductions. Now again, if we just bring these two data sets together and think about what they collectively mean, we can see that all of our zebrafish are moving. It's just that those exposed to both UPM and cortisol are moving less in some instances. What do these changes in behaviour mean for neurodevelopment though? Well, we're unsure. There's a lot of possibilities around this. For example, it could be vision-based, say if the UPM is affecting the eye development in some capacity. This would naturally have a knock-on effect for overall movement, but again, there are many possibilities which warrant further investigation. To investigate structural changes, we can start to look at different imaging techniques. These images are important in of themselves though, because we can compare different regions of the brain between individual zebrafish just by looking at them. And so here's an example using the midbrain from a zebrafish sample raised in system water, and then another which was raised in UPM, exposed to UPM only. Now, while there are clear differences between the two, we can tell that, morphologically speaking, the gross neuronal architecture remains largely unchanged. And this is perhaps yet another suggestion, another sign that the changes we are seeing in behaviour and morphometrics is down to much more subtle reasons. But moving on to the computational side of this section of my work now, the first step when working with images usually involves converting them into grayscale images. That way we're working with a single colour channel instead of the usual 3, which is RGB. This means each image now consists of pixels which have values that lie between 0 and 255, 0 being completely black and 255 being fully white. Note that this step is not needed for our GFP confocal images that I just showed you, because if you remember those cases, we're only using the green channel, and so that means that only the green channel has pixels with non-zero values, and so essentially it's the same thing as a grayscale, except instead of it being grey, it's green. The only reason I'm showing you this now, however, is because this overall concept of converting images to grayscale is very important for the general pipeline if we were to work with images that were RGB, for example. And secondly, we want to then segment our images. This is another common step, and it's important for removing background noise. And what is that? Well, this is data that we don't want. So pixels which, if we left unchanged, would affect our computational analysis in really unhelpful ways. What we actually do is use Otsu's method, which separates every pixel into foreground, which is what we want, and background, which is what we don't want, or the noise, as it's more commonly referred to. And so, for example, say if that threshold was deemed to be 65, then every pixel equal to or less than 65 would be set to 255, so fully white, which is what you can see in the image on the right-hand side, and everything above 65 would be left unchanged. Now, for the sake of this presentation, we actually hand select the region of interest to be that which contains the intertectal projections. And these are the branch structures that you can see in the center of the midbrain. This is useful to test new computational techniques on, because firstly, we can see that there are clear structural differences between the two images, if you look at A and B over here. And secondly, this region also has neurological significance. The midbrain is believed to be responsible for learning and play a key role in optic connections. And if you remember from earlier on in the presentation, this may be linked to why we're seeing changes in behavior and morphology. To analyze these branch regions in the midbrain, we borrow from an emerging and highly advanced field of computational mathematics, which is referred to as topological data analysis. And in particular, we use persistent homology to investigate features within our confocal images that relate directly to the topology. So this is its shape, it's its structure. And this gives us really valuable information regarding the number of connected components and the number of holes. And I'm showing you the results regarding these on the screen over here. Now, the biggest question you might be asking right now is what on earth is a connected component? What on earth is a hole? What do those words even mean? Now, they are quite tricky concepts to explain in general, not least in this condensed format. So let's take this example image that I have here of Durdle Door. We can see that this is an RGB image, and so we would convert this to a grayscale in the same way that I've described, and that would give us thousands and thousands of individual pixel intensity values between 0, which is black, and 255, which is white. And just looking at this image in particular, we can see that there's sort of spots of white around here. We'd expect very high values, and then these sort of darker ridges on the mountains and the rocks, we'd expect those to be much lower values. And so connected components can be thought of as clusters of dark pixels that sit against a background of brighter pixels. Holes, on the other hand, exist when we have brighter pixel regions that are encased in darker pixels. And so, for example, lots of bright pixels dotted sporadically against a very dark background would equate to lots of holes with one or very few connected components. And so now let's take the concept of the image that I've just shown you here and scale up. Instead of each pixel being a square with a number inside, imagine those numbers projected as bars, where the height of the bar is due to the individual pixel intensity value. So over here we had lots of white regions where the sort of C was. We would expect those to be very, very high bars because it was a very high pixel intensity value, close to 255. And then here in the center of the image where we had the ridges, the darker rocks, we'd expect those bars to be much lower because they were close to 0. And then what I do is take vertical slices for each pixel intensity value, highlight the surface below it, and work out the number of connected components and holes within that surface. And through that method, you get the sort of data that I'm presenting on the screen right now. And so this slide just serves as a visual pipeline for each of the steps that we've just walked through. So hopefully you can get a better understanding of how the two different confocal images, which is what we had originally, and you can see those on the far left, how they lead to different interpretations when we use persistent homology. So to conclude everything, does exposure to air pollution and/or other stressors, in this case cortisol, have an impact on neurodevelopment? We think that it does, and the work that we've completed so far is suggesting this in some variety. Both behavioural data and morphometric measurements indicate that there are differences between embryos exposed to UPM and cortisol compared to our controls. Reduced movement and velocities when tracking behaviour and changes in head measurements were also detected as well alongside all of this. And we also did see notable visible changes to the neuronal architecture, which we then further analysed using methods in topological data analysis and persistent homology. But we don't just want to look at topological data analysis on its own. We also want to try additional methods such as machine learning, and that would play a part in our computational analysis and the new methods that we'd look at there. But perhaps the main thing that we want to look at going forward, at least immediately, would be some H&E staining that was recently done by Molly Mann, who is an MSci student in our lab. In this example that I've got on the screen now, we have a lateral view of the midbrain, which is the same region of the brain that we've been looking at through our GFP confocal images in the presentation. And we can see here that there are clear cellular morphological differences when exposed to UPM compared to the control. But interestingly, we didn't see this same phenomenon in other regions of the brain when exposed to UPM as well. So this indicates that there's a lot more going on here, and this is something that we want to investigate further. And then finally, I just want to finish off this presentation by thanking everyone involved who is behind the scenes. I myself am part of the McGonnell Lab at the RVC, but various academics from different departments have given really valuable insight and helped with technical work along the way. I especially wanted to thank Molly Mann for her contributions with the H&E to my project, and also Moise Azif, who is actually a current PhD student at the University of Bristol, for all of his help regarding implementing and interpreting our persistent homology data. On top of that, you can find a list of the key references throughout this whole talk in the description below. Thank you very much for following along, and I hope you found it really interesting.